One modern cancer research paradigm holds that understanding the genetic alterations leading to cancer will lead to improvements in prevention, early detection, identifying prognostic markers, and developing targeted therapies. This oncology database in Microsoft Access spreads across 7 primary tables, including a patient's table with clinical information, a TMA table with protein expression data, and a DNA specimens table with mutational data. Relationships are developed to link the data among the tables so that the laboratory studies of tissue belonging to a particular patient can be connected with his or her clinical history. The linked data can then be retrieved via a query of the database and statistically analyzed to determine relationships among protein expression, DNA mutations, and clinical outcomes. Ultimately, linking clinical and laboratory data facilitates the study of genetic aberrations and protein expression from the perspective of clinical endpoints in cancer patients.
A key component of translational research is the ability to accrue clinical and basic science research data and amalgamate the two in a meaningful relationship. However, the bioinformatics tools necessary to do this are often limited and institution-dependent.
This database was developed to help our researchers answer key questions relevant to translational cancer research, such as how a patient's genetic and proteomic makeup can contribute to certain clinical endpoints, such as response to therapy or survival.
Though this database was developed for researchers and clinicians involved with thoracic malignancies, this model can be applied to other diseases and to suit the needs of researchers and other institutions. We hope